I just want to formally welcome you to this Ocean Carbon and Biogeochemistry, or OCB, program activity. OCB is a coordinating body that focuses on activities relevant to carbon cycle science in the ocean. And we are really about forming a network we support a very large network internationally, actually, of scientists who work across disciplines to examine the ocean's role in the global carbon cycle, looking at changing biogeochemistry and marine ecosystem function and marine ecosystem change. And this community has been, the OCB community has been really heavily involved in pushing the Arctic Colors campaign forward. The planning for this campaign has been ongoing for a few years now, and this is an activity that was selected by OCB in the last activity solicitation, and it's really focused on synthesis. So we're going to spend the next 3 days looking first at field observations, and then we'll be looking at modeling capabilities, and we'll be then looking at satellite remote sensing capabilities and how we can gather what we know to set the stage for moving forward with a large-scale field campaign. So we are really excited to be supporting this activity, and I'm going to hand it off to the organizers. I believe, Maria, you'll be speaking first, leading us off, and then we'll proceed with the remainder of the talks.
Yes, thank you so much, Heather. Let me share my screen. Great, can you see my screen?
Yes. Yes, we can.
Great, so hello everyone. Welcome to this open virtual community meeting. Thank you so much for joining us today. I'm Maria Giorgio, professor at City University of New York and one of the lead PIs for the Arctic Color Scoping Study. Co-organizers of this 3-day meeting are Antonio Manino at NASA Goddard, Patty Matre at Bigelow, Peter Hearns at UC Davis, and Robin Spencer and Nan Kellerman at Florida State University. Now, this meeting is being organized as a pre-Arctic Colors data synthesis activity, and we're really, really grateful to NASA's Ocean Biology and Biogeochemistry program and the Ocean Carbon Biogeochemistry Project Office for sponsoring this activity. Heather, many, many thanks for your help, not only for sponsoring this activity, but also for supporting Arctic Colors meetings and workshops over the past more than 6 years. So what is Arctic Colors? Arctic Colors is a field campaign scoping study selected by NASA's Ocean Biology and Biogeochemistry program to design an interdisciplinary oceanographic field campaign program that addresses high priority science questions related to land, ice, ocean interactions in the rapidly changing coastal Arctic. You see here the annual Arctic sea ice minimum area. 2020 was the second lowest on record. The 2021 minimum just a month ago on September 15th was not as bad. It was the 12th lowest in the nearly 43-year satellite record. But the last 15 years have been the lowest 15 sea ice extents in the satellite record. So with this group, I don't need to try to convince you why more measurements are needed in the coastal Arctic. We all know the land and the ocean in the Arctic are changing rapidly, with important consequences, of course, for Arctic wildlife and human populations. On land, permafrost is thawing. In rivers and lakes, ice-free periods are getting longer. River flows are changing. The Arctic Ocean is warming, is freshening. Sea ice coverage is declining, but also sea ice seasonality is changing. Primary productivity is changing, and we're seeing an expansion of harmful algal blooms within the Arctic. Now, there have been ground-based monitoring programs focusing on measuring these changes. The problem is that most of these monitoring programs have been focusing either on land or the ocean primarily. And as we will be discussing today, we still know very little about processes, interactions, exchanges, and feedbacks across the Arctic land-ocean continuum, and the impacts of these processes and feedbacks and exchanges on ocean salinity or primary production, biodiversity, sea ice formation, and larger scale ocean circulation patterns. So what makes things more complicated is that the Arctic coast is difficult to access. So existing data sets have very limited spatial and temporal coverage. Satellite remote sensing can provide a different perspective, an integrated coastal Arctic system perspective that we cannot get with surface measurements alone. So this perspective is partly what has driven Arctic Colors, based largely on remote sensing observations from space, but also integrating measurements from other platforms, as well as models. Arctic Colors aims to quantify the coupled biogeochemical ecological response of the Arctic nearshore system to changing terrestrial fluxes and ice conditions. And we want to do this in the context of environmental short-term change, but also longer-term climate change. Now, our community has been working on the Arctic Colors concept since 2014. Over these years, we have developed a science plan with input from the broader community, some of you here with us today. We worked on outreach materials and we built a science team with international participation that has contributed significantly to identify high priority science questions and also try to link to other field activities in the Arctic. And you will see some of this information, some of these activities with the first presentation today by Kellerman. As NASA prepares for this large-scale field campaign, an important step is to identify and characterize existing field data sets, field observations, as well as modeling activities and remote sensing capabilities that could be applied to the coastal Arctic area that is relevant to the Arctic Color study domain. So the main objective of this 3-day meeting today in the next couple of days is to introduce this phase 1, this pre-Arctic Colors data synthesis activity, and as a community start discussing how we can share this information, how we can share information on past, current, and planned field campaigns, as well as modeling and remote sensing activities. In early 2022, we would also like to have what we think would be a hybrid workshop where anyone interested in sharing and contributing existing data sets, models, and algorithms for the Arctic Color study would participate in this data synthesis and combine data analysis. We have an opportunity to participate in writing of overview manuscripts and other reports. So this virtual meeting today is really a very important step toward the development of the Arctic Colors implementation plan, really, and eventually the Arctic Colors field campaign itself. Now, a campaign of the scale of Arctic Colors requires a multidisciplinary, multi-method, integrative observational approach. As we describe in the science plan, there is a need for a diverse array of measurements, measurement approaches, so that we can have year-round observations and sampling. Satellite remote sensing, of course, is central here, and there will be a range of sensors we could use, not only ocean color, but multidisciplinary data to address the Arctic Colors science objectives. And also coupled land ocean ice models are critical here for studying biological and biogeochemical processes, for resolving observed seasonality, for resolving variability in terms of spatial variability, but also assessing relative importance of different processes, as well as impacts on ecosystems. So this synthesis needs to happen across these data sets, for field observations and in situ data, as well as modeling frameworks and remote sensing aircraft and satellite algorithms. This is why we're having a 3-day meeting. Day 1 of this workshop will focus on relevant field measurements, including large field campaign programs. Day 2 will focus on modeling activities with presentations highlighting some major modeling efforts in the coastal Arctic. And day 3 will focus on remote sensing, discussing progress and challenges, as well as what are some of the most critical needs in terms of satellite and airborne remote sensing algorithm development, data records, and data products. Today, we will start with an introduction to Arctic colors and discussion of relevant large field campaign programs and collected field observations. Specifically, we will have some updates from the Arctic Great Rivers Observatory, the Beaufort Lagoon Ecosystem Long-Term Ecological Research Site that was funded by NSF in 2017 to study lagoons and other aquatic systems along the northern Alaskan Arctic coast, as well as the MALINA and Nunataryuk projects. After a short QA session for all speakers, we will move to a 45-minute discussion in breakout groups. Each breakout group will be addressing the same questions, but we just wanted to have the discussion in smaller groups to facilitate discussion and exchange of ideas. And we will then come back to the main room to report on the subgroup discussions and discuss as a whole group. Now, as you're thinking about Arctic colors and potentially participating in the future in this major field campaign, please feel free to contact any of us, any of the scoping study PIs and organizers of this meeting. Our emails are here, or even better, feel free to contact the NASA OBB program scientist Laura Lorenzoni to learn more about Arctic colors. So let's start. Our first presentation will be an introduction to Arctic colors and the OCB data synthesis activity by Anne Kellerman, postdoctoral research at Florida State University. Anne, the floor is yours.
Thanks, Maria. Hi, everyone. Maria, you made that really easy for me, so I can go through some of this a little bit quicker. And yeah, I just want to also say welcome, everyone, and thank you for coming. So this presentation is about a little bit more about Arctic colors, and then also the coastal Arctic biogeochemical data synthesis activity, which we're here to talk about today, the next 3 days. So first, an overview. Oops. Where did that go? An overview of Arctic colors. What is it? Why it matters? What's planned? And then a description of the OCB data synthesis activity. Sorry, I just need to move this out of the way. Yeah, whatever. Okay. Okay. There it is. Okay. So the Arctic color summary. Arctic colors stands for Arctic Coastal Land Ocean Interactions. Arctic colors is, as Maria said, is a proposed NASA field campaign to examine the effects of land, ice, and future change on nearshore Arctic biogeochemistry. But first, some background. I'm just going to touch a little bit on a changing Arctic and rivers as integrators and transporters of organic matter and nutrients. And if you'd like more information, feel free to visit the website, which is arctic-colors.gsfc.nasa.gov for more information. So we all know that the Arctic is changing, and everyone has seen a figure similar to this where it shows that the Arctic is changing disproportionately to the rest of the globe. 2020 was again the hottest year on record, and July 2021 this year was the hottest month on record ever. This has implications for many things, including the decreased sea ice extent and duration of color, decreased landfast ice, increased coastal erosion, and increased permafrost thaw. Maria had a lot of really beautiful pictures illustrating a lot of these things. And that has impacts downstream on a lot of things, including the lives and livelihoods of human populations, of folks that live in those areas. And it also has an impact, or the alteration of land-ocean transport and processing of organic matter and nutrients. The location of the Arctic colors study domain is in the Yukon, is basically includes the Yukon and Mackenzie rivers and the coastal area in between them. And then also the near shore environment. The Mackenzie and the Yukon are the 4th and 5th largest Arctic rivers by annual discharge, 18th and 22nd globally, with mean annual discharge of 316 and 208 cubic kilometers a year, respectively. And both of these rivers are largely underlain by permafrost. This is a good place to be doing this this research because there are several notable events in and around Alaska in the last decade, and this is just a figure showing a lot of notable events that happened between 2014 and 2019. So, for example, you had 5 warmest winters in Nunataryuk, longest growing season, record heat in different areas. So this change is happening in Alaska and the Mackenzie River Delta, and it's happening now. So the time is now. Why rivers? 10% of Earth's flowing freshwater discharges into the Arctic Ocean. Rivers modulate the land to ocean transport of organic matter and nutrients. Rivers integrate inputs and signals from the catchment. And changes in export and processing will have implications in the nearshore coastal zone. But rivers are also important to illustrate what's happening with changing freshet. There's changing patterns in precipitation and temperature, and those are altering the timing and dynamism of freshet. This will likely have implications for the amount and the timing of land to ocean carbon transport. And we need to set a baseline of current conditions to understand the extent of future change with Arctic colors. So Arctic coastal land-ocean interactions, Arctic colors, is a proposed NASA field campaign program that aims to quantify the coupled biogeochemical and ecological response of the Arctic nearshore system to rapidly changing terrestrial fluxes and ice conditions in the context of environmental and climate change. The overarching science aims are to establish the effects of land, ice, and future change, including warming land and melting ice on the nearshore Arctic biogeochemistry in this area. And the figure to the right is a map of the study domain. The pink area in the coastal waters shows the nearshore coastal environment that we're particularly interested in, along with the different rivers in the region. So a little bit more specifically, we want to look at what the effect of land on nearshore Arctic biogeochemistry is. So how do freshwater, carbon, nutrient, and sediment fluxes to the coastal zone change as a result of several things, including changing riverine and groundwater inputs, passage through estuaries and gradients. So we kind of know that the nearshore coastal zone is seen as this biogeochemical filter. So there's a lot of activity going on there. Coastal erosion and thawing permafrost. How do these changing fluxes affect nearshore Arctic biogeochemical and ecological processes? How has the relative magnitude of inputs from rivers and coastal erosion changed across the nearshore Arctic seasonally and interannually? The next question is, what is the effect of ice on nearshore Arctic biogeochemistry? So how does flow alteration and channeling by morphological ice conditions impact terrestrial fluxes into and attenuation within the nearshore Arctic? How does the coastal snow and ice cover impact nearshore Arctic biogeochemical processes by controlling rates of mixing and by modulating light availability? And how does the timing of sea ice formation, retreat, the timing of sea ice formation and the timing of sea ice retreat, duration of sea ice cover and ablation, snow accumulation, and the morphology of the coastal ice zone influence nearshore Arctic biogeochemical and ecological processes? We want to know how the effect of future change, including warming land and melting ice, on nearshore Arctic biogeochemistry. So we want to essentially asks these questions on different timescales, including seasonal timescales, interannual timescales, and interdecadal timescales of how will changing land and melting ice impact nearshore Arctic biogeochemical and ecological processes. The Arctic Colors Science Plan includes integrating coupled land ocean ice models with remote sensing data and field campaign observations to forecast and hindcast carbon fluxes and productivity across the land sea ice continuum. It includes implementing emerging satellite and airborne remote sensing data fusion capabilities to integrate across multiple spatial and temporal scales. And then also utilizing field campaign observations to evaluate and improve NASA satellite ocean color retrievals in the coastal Arctic. For the reason we're here today, the Coastal Arctic Biogeochemical Data Synthesis activity. The aims of this activity are to provide a comprehensive synthesis of existing ocean biogeochemical data in northern high latitudes. Primarily, we're looking to synthesize data on carbon stocks and fluxes along with optical proxies, for example, absorption and scattering, and any appropriate metadata. Other biogeochemical variables such as nutrients and processing will be included where possible. We're also looking to collect and combine existing in situ data, modeling results in satellite data sets, resolve differences in reported data due to methodology, create and host a newly synthesized database, and identify data and knowledge gaps. The notional timeline for Arctic Colors looks like this, and you can see that it builds on the establishment of a number of programs that are already in place, including the Arctic Boreal Vulnerability Experiment, the Beaufort Lagoon LTER, which Jim McClelland is going to talk about later. There's a number of pre-Arctic colors activities going on already, and we are in this, and as well as the OCB data synthesis activity with the solicitation phases coming in the future. So the aims of this meeting are to bring together all of you, so scientists working on land, ice, ocean processes, and across the rivers to coastal ocean continuum within the Arctic Colors study domain and potentially adjacent land and ocean systems, to identify existing field measurement data sets and planned field activities relevant to Arctic Colors, to confer on the status of existing models pertinent to Arctic Colors and identify challenges in advancing those modeling efforts, to identify existing remote sensing capabilities, including airborne data sets fine-tuned for the coastal Arctic, discuss activities that would reduce uncertainties in satellite and airborne remote sensing data products, and to facilitate a compilation and preliminary analysis of existing field data sets from the Arctic Colors study domain, resulting in a public data repository and at least one manuscript. So with that, I'd like to acknowledge a bunch of people, a lot of the same people that Maria acknowledged already, but I think it's really important to acknowledge all the people that have brought Arctic Colors and the OCB data synthesis activity this far, including Heather Benway in the Ocean Carbon and Biogeochemistry Program, Paula Bontempi, Laura Lorenzoni in the NASA Ocean Biology and Biogeochemistry Program, the NASA Review Committee that reviewed and provided useful feedback on the science plan, and participants of the 2016 Arctic Colors workshop at Hui, all the additional Arctic Colors scoping study PIs and collaborators and co-authors. And then I'm just gonna put this up briefly. I know it's a lot of text. So these are some of the questions that we're gonna provide for the breakout discussions. And I don't expect you to read all of it. I'm not gonna read all of it to you either. But basically, we're just interested in, that the discussions could focus on areas, and I think it's important to have just a little bit of the questions ahead of time before the presentation so you can sort of start thinking about them during the presentations. But based on what we hear about soon, what areas along the Arctic colors domain have the richest data sets, and some that, and which areas have very little information for addressing the Arctic colors objectives, which data sets are available already, what are some challenges that you've experienced or foresee in reaching these objectives, and then what are particular successes that colleagues have had with promoting and amplifying Indigenous communities and collaborators. And then the last question is more for the activities tomorrow and Wednesday about, I actually can't see it anymore, but about the upscaling of modeling and remote sensing. Sorry, my video images are in front of the bottom question. I can't see it, but maybe you can see it, and hopefully you'll have a Google Doc access to a Google document with it on it later during the discussion. So with that, I believe the next speaker is Rob Spencer, also from Florida State University, to talk about the Arctic Great Rivers Observatory.
Hello, everybody. Hopefully you can see my screen there. And I apologize, I have somewhat of a cold, just a regular cold. And so I apologize for being a little bit croaky today. So I was asked to talk today about the Arctic Great Rivers Observatory, which is a long-term multi-river observational platform now that's been based out of a number of institutions that you can see listed there, from the Woodwell Climate Research Center to Florida State, University of Texas, University of Alberta, and has received funding from NSF. I've put the web link on that first page, but I'll come back to it at numerous times throughout, as one of the real important things I wanted to mention today is that all the data we generate is freely open and accessible and there for the community to use. So as an outline of my talk, I'll talk briefly about some history on the Arctic Great Rivers Observatory. Anne's already covered this, and so did Maria, about why we care about Arctic rivers, and I feel like I'm sort of preaching to the choir here today, so I won't spend too long on that. But I wanted to come back and talk a little bit about characteristics of the Pan-Arctic watershed and some of the challenges in terms of scaling, and how that's of relevance to Arctic colors, where we are in terms of our understanding of current fluvial fluxes, and how we're now able to start looking at the signal of change in this multi-decadal data set that we have for the Arctic Great Rivers. So for those of you that might not be so familiar with it, this is how I often like to think about the world, looking down on the North Pole. And so we're talking about the 6 largest Arctic rivers: the Ob, the Yenisei, the Lena, and the Kolyma in Russia, and the Yukon and the Mackenzie in North America. And so this began as what was the Partners Project back in 2002, which stood for Pan-Arctic Riverine Transport of Nutrients, Organic Matter, and Suspended Sediments. And one of the big drivers for this, which is obviously something we need to be aware of as we think about Arctic colors, is standardization of protocols across these different river basins in this case, driven by issues like if we're comparing, say, nutrient data sets from the Russian rivers to nutrient data sets from the North American rivers, where there was historically different analytical methods, how comparable were they in terms of looking at long-term drivers and change? And so all samples are now analyzed together, and samples are collected at multiple times of the year and all phases of the hydrograph. And as this project's progressed, there's been different sort of sampling schemes in trying to catch that. There's been a focus at times really to nail down the freshet, but now it's a very standardized every other month sampling to try and get equal samples representative of under ice conditions through peak discharge in the spring, and then in those open water summer months as well. As I mentioned, and again, I really wanted to push this. All this data is available. Please go to the Arctic Grow website, and that goes back to all the data sets that exist from historically partners in 2003 to 2006 through to Arctic Grow 1. So there was a small break there for a couple of years, which ran from 2009 to 2011 in terms of data collection. Arctic Grow 2 from 2012 to 2016. Arctic Grow 3, which just finished in 2020, and we're currently through into Arctic Grow 4, which is funded out till 2024. So there's biogeochemical data there on those 6 rivers that I mentioned already, and there's actually daily discharge data for 15 Arctic rivers. So those large 6, the big 6 if you like, and then 9 other rivers in Siberia. Again, thanks to NSF for supporting this, and for those of you that might be interested, there's the web link, and it's pretty easy to find as well if you don't have a chance to write that down, and if you Google Arctic Great Rivers Observatory, you'll find it. On the website, just to give you a bit of an overview of what's there, you can find access to all the data sets split out into what we think makes sense for different users. And so of interest to this community might be all the absorbance data that's there, along with the discharge and water quality data. There's also some spatial data that's available for these big 6 rivers. And this is kind of what you get if you go into the water quality data. It's all broken down by each individual river basin and is available to be downloaded. We try and update this as soon as possible. As soon as data is available, it's updated and put on the website. And so please check back frequently for the most up-to-date data. And then what we sort of do with this is track where it ends up in terms of publications, and we classify those into a number of different realms, from the primary publications driven by the core Arctic Grow PIs or project personnel, through to what we're seeing, and obviously very happy to see, a lot of secondary and tertiary publications coming out where the community is taking and using this data in a host of different ways. So as that hopefully gives you a little bit of background about the project, but what are the motivations for this? Why do we care so much about Arctic rivers? Thanks, Anne, for already touching on some of these, but we have a very small ocean in terms of the Arctic Ocean basin size, less than 1% of global volume. We have over 10% of global river flow coming into it. So we have a very small ocean, a lot of large rivers coming in. So there's a lot of motivation for improving traces. We've done some work historically on things like, say, lignin phenols in the organic geochemistry realm to try and get a better handle on that land-ocean flux. And I'll talk a little bit about that today, as well as Anne mentioned, to interpret processes and changes that are already occurring throughout those watersheds. And those changes are already underway. We've seen increasing river discharge for a long time now, active permafrost thawing, as well as shifts in vegetation in these major basins. So for the purpose of my short presentation today, I chose to focus on this as the Pan-Arctic watershed. There's a number of different ways which you can slice this, and happy to chat about that with people. But this is basically a working sort of Pan-Arctic watershed where all the water that's coming off the land, off the terrestrial landscape, is ending up back into the Arctic Ocean. And you can see it's dominated, the vast majority of it in Russia, about nearly a quarter in Canada and the US. But you see this fraction as far down as places like Kazakhstan, equivalent to what's draining in from the US, because these large, particularly Siberian rivers, are draining far down into the Asian steppe. And so along with that, and I'll hopefully convince you of this, they don't necessarily have the most Arctic characteristics for some of their drainage basins. If you ask many people what they think about in terms of Arctic vegetation, they'll say tundra, and only 15% of this Pan-Arctic watershed, which you can see there highlighted in green, is tundra vegetation. If you look at permafrost, it's about 45% is continuously underlain. You've got watersheds like the Kellerman that are 100% underlain with continuous permafrost. As Anne noted, there's a mixture in the Yukon and the Mackenzie, and then there's basins like the Ob that have a very small permafrost coverage in their basin. So there's large differences between these rivers. So all of this becomes an interesting question when you start looking at scaling. And I think this is one area where there's a lot of interest in Arctic colors in terms of how we can improve scaling. And so if you take that Pan-Arctic watershed, those big 6 rivers are covering about 2/3 of the area and about 2/3 of the discharge as well, with the next 8, the middle 8 rivers there that are all in Siberia, only giving you an extra 15% and the remainder giving that extra 20% there. When you look at tundra, you see that those big 6 rivers are not very representative, for example, in terms of vegetation cover, when you compare to those more northern constrained higher latitude watersheds. And you see a similar thing when you look at, say, percent continuous permafrost, where the Pan-Arctic watershed has this big range from the Kellerman through to the Ob, from 100% to 2% underlain by continuous permafrost. But overall, the big 6 come out at about 35% underlain by continuous permafrost, whereas those smaller northern higher latitude constrained basins are unsurprisingly typically more underlain with a much higher continuous permafrost content. So just a point there on scaling, and I'll come back to that. With this project having run for a number of years now, I would say we have a good handle on current fluvial fluxes. Again, all this data is freely available for the community to please go and utilize. And you can set up nice, in this case, the plot on the left, discharge concentration, relationships between whichever river you're looking at. And if you look there in that bottom left-hand panel, when you have high discharge in the LANA, unsurprisingly, you have high DOC concentrations, and that means that over about half of your DOC flux is occurring in that short 2-month period associated with the spring fresh air. And so it really shows the importance of capturing a good even spacing across the hydrograph, so that you know that you can convert this into good data in terms of seasonality, seasonal flux of these analytes to the Arctic Ocean. One thing I thought I'd mention of interest to this community is there's a huge database there of CDOM measurements. Obviously, they allow for rapid and relatively inexpensive monitoring of DOM. It can be done from remote sensing techniques. It can be done in situ. And we found a pretty strong relationship between CDOM and both DOC in the top left panel here and lignin concentration, vascular plant biomarkers, so great trace of terrestrial material coming into the Arctic Ocean. We can do that in situ now, potentially going forward, and it gives us huge potential to improve flux estimates using things like CDOM into the coastal ocean, and then to track its fate into the coastal ocean as well. So using those new estimates, where we had a lot of data for CDOM and a limited data set for lignin, it's analytically intensive, it's expensive, and you need a fairly large volume of water, certainly compared to what you need for CDOM to make a measurement. We estimated new DOC loads, which are sort of in line with what other people are estimating now, but I always like to remind people it's about 50% more or so than we were estimating only a decade ago. But the lignin flux increased dramatically. It actually doubled from the historical lignin load. And that's primarily due to capturing that highly dynamic part of the hydrograph with daily CDOM measurements across it in that spring period. Obviously, if you have a larger flux of lignin to the Arctic Ocean, that means that terrestrial DOC is transiting through even faster, and your residence time is going to go down. And so it's highlighting that Arctic River and DOM is more reactive than previously thought. And all that comes from a simple improvement in flux calculations, pairing high resolution CDOM measurements, which we can still, I think, continue to improve, to geochemistry biomarkers like lignin. On the DIC side of things, and this is where I'd say we're kind of making the most progress at the moment for detecting long-term signals of change. We've been looking at alkalinity. So that's sourced from chemical weathering, represents a sink for CO2. And alkalinity flux in these watersheds is controlled by a host of things like precipitation, permafrost extent, groundwater flow paths, and surface vegetation. And all of those are impacted and all changing due to warming in the region. So across the past 35 years or so where we have data, so this is taking the Arctic Great Rivers Observatory data set and putting it together with a data set from the mid-70s through to early 90s from the USSR, we find that we see large increases in alkalinity flux for the NSA at about 185%, and for the OB at about 134%. And if you look in both cases, so this is the OB data here, the NSA data here, I hope you can see my cursor, you see these increases in alkalinity concentration, and the NSA has also seen an increase, a pretty dramatic increase in discharge, whereas you don't really see that in the OB, which is accounting for some of that large difference in export that we get between them. What's causing this? Well, all of the above. We're diving deeper into the data now to try and tease that apart. But they're all linked to temperature as well as potentially decreased acid deposition in the region as well. So if we look at the relationships with temperature, And you can see here the mean annual temperature anomaly across latitudes relevant to these watersheds. You see concurrent with that increasing alkalinity export with increasing mean annual temperature anomaly. That obviously has implications for the rate of carbon sequestration on land and delivering of that buffering capacity to coastal environments. And I also think this is incredibly useful because it gives us the ability to project moving forward with model temperature rises for the Arctic ecosystem, assuming we're seeing a fairly linear response in the system here. So I hope you could understand me. I sound very croaky in my own head and I apologize for that. But the main points I think I'd like folks to take away from this today is that, you know, much of the watershed, particularly in terms of these big 6 Arctic rivers that drains into the Arctic Ocean, isn't Arctic. So we have to be cognizant of that in terms of upscaling. The contemporary baseline for those big 6 is now really pretty well established. I think we're fairly confident in our export numbers for a whole host of analytes. And so we're now starting to look for that signal of long-term change. And interestingly, we're seeing it quite nicely in some of the data, as I highlighted in the alkalinity data set there. Extrapolation to the Pan-Arctic is still problematic. Again, getting back to this scaling issue. And it's one of the strengths I see here with connecting well with Arctic Colors, with long-term projects like Arctic Grow, where we can compare some of our data sets to proposed data sets in some of these constrained northern high latitude ecosystems. And I'm sure Jim McClelland's going to talk about that in a second. And just to highlight, and everyone knows this, but I think we've done a pretty good job of getting or estimating that flux to the head of tides, to that first where we hit the freshwater seawater interface. But that's very different to what's making its way out to the ocean. And so connecting as a community across that boundary, from those of us who focused on riverine-based studies to those who are more on the marine end, I think there's a lot of progress that can be made here into the coming years. And so I will stop there and hand over to the next speaker, who I believe is Jim McClelland.
All right, let me just see if I can get organized here. Can everybody see that? I guess I see at least one nodding head, so that's good. Yeah, so I'm going to spend a few minutes talking about the Beaufort Lagoon Ecosystems Long-Term Ecological Research Project. I probably have more slides than I can get through in 15 minutes. So what I'm going to do at the front end is set up so I can give an overview of the BLE LTER completely, and then I'll sort of circle back and talk about some more things in greater detail, time permitting. But I'd like to start off by just acknowledging that this is a large collaborative project. It involves 12 PIs from 7 institutions. There are currently 12 students involved, graduate students, and some very hardworking and dedicated project administrators serving in various roles that make this thing work. We also have some really important partnerships with various organizations, including the U.S. Fish and Wildlife Service, USGS, BOEM, and the North Slope Borough School District. Okay, so yeah, so the BLE is part of the National Science Foundation LTER network. There are 29 LTER programs currently, 4 in Alaska, 11 coastal and marine. We're the only coastal, well, near shore coastal in the Arctic. As was already mentioned, we got rolling in 2017 in terms of when the funding came in. Our first field campaign was in the summer of 2018. If you boil down what we're doing to one overarching question, it really comes down to how do interactions between terrestrial inputs, ocean exchange, and sea ice dynamics influence Arctic lagoon ecosystems over seasonal to multi-decadal timeframes. Obviously, there's a very important climate change overlay to all of this, and but the LTER program in general also really emphasizes fundamental ecological processes and we're using these systems to test and their dynamics to test ecological theory in various ways. So yeah, in terms of the terrestrial input side of things, we're talking about coastal erosion, watershed runoff, groundwater inputs. And in terms of the ecosystem properties in the lagoons, biological production, community composition, food-web relationships, and biogeochemical cycling more generally. We work out of 3 different locations: Utqiagvik, Deadhorse, and Kaktovik. We refer to these different locations as nodes, and then we have a couple of focal sites or lagoons at each out of each node that we work. We're doing we have in situ sensors that are gathering data year round. The project includes some remote sensing work, particularly as related to coastal erosion aspects of the project. We make major field campaigns 3 times a year, and then have hydrologic modeling and ecosystem modeling components that tie things together. This bottom sort of schematic, the red stars on here show when our field campaigns are, but in general, the idea is we get out there at the end of winter while it's still ice covered. We get out there shortly after breakup, or during ice breakup, shortly after the big sort of spring freshet pulse of runoff. And then we get out there in the open water period later in the summer. And although not shown on that last map, this includes some sampling of streams and rivers at places that we visit repeatedly. We have three at the western node: Avok Creek, Mayo River, and No Name River. Ironically, the Avok Creek is considerably larger than either of these things that are called rivers. But at the central node, it's the Kaparik, the Segav-Nurtuk, and the Put River. Probably the only 2 of these you all recognize would be the Kaparik and the Sag. And then the eastern node, we've selected the Jago River as a focal site. Yeah, so I mean, so why these rivers? I guess mainly we're leveraging previous studies and building on existing data sets. We of course want those data for flux estimations in the proximate sense. We're also sort of acutely aware of the need to develop modeling capabilities, and those provide at least some points of reference for calibration and validation. In terms of core program measurements, I'm definitely not going to go through these in great detail, but the point here, I guess, is we're measuring a great many things, some of them continuously. Over here, other things that we do during these field blitzes, which really sort of that these 3 columns focus on, and I think many of which are relevant to different aspects of Arctic colors. Okay, how am I doing for time? Good. Right. So that was, I guess that's the broad overview. I'm going to dig into a few data sets in greater detail. So one of the things we've done at the front end of the LTER is try to pull together the data that we had in hand and do some synthesis activities. One of the things that emerged from that by pulling together data from a bunch of stream and river sampling on the North Slope, and you'll see also in here in white the 6 big rivers that Rob just talked about. All the other ones that are colored here come from the North Slope. But anyways, we looked at dissolved organic matter concentrations, DOC and DON, as a function of a variety of watershed characteristics. But what emerged from that is, in particular, is that there's a really strong relationship between watershed slope or steepness and the amount of organic matter in the water. And that creates a nice, at least first cut tool for estimating concentrations from drainage areas that we can't actually visit. When we apply that across the North Slope domain to all of these drainage areas that are colored in here north of the Brooks Range, one of the interesting things that emerges is that there's a pretty strong west to east gradient or decrease from relatively high concentrations of DOC in the west and decreasing to the east. And that has some interesting implications for sort of potential sort of trophic drivers and ocean color in general, I would say. We're doing, so that was an empirical approach to get at the sort of estimating concentrations of stuff where we don't have sampling going on. We're also developing models. I won't talk about this now because Mike Rollins is going to spend time talking about his modeling efforts tomorrow. But I will say that we recognize that across this large domain we're working, that ultimately to tie things together, we really need the modeling component. And Mike isn't only modeling watershed sort of fluxes of water, but he's made good progress on estimating DOC fluxes through his modeling efforts. We're doing some groundwater work. That's something that gets less attention in the Arctic, but that we, at least at the scale of these lagoons and really close to shore, becomes interesting to think about. And I don't want to spend a lot of time on this, but I'll say that we sort of, what we're talking about here is supra permafrost groundwater. In other words, water that's moving through the active layer, above the permafrost and making it into the lagoons, having not seen the light of day. And that then creates an interesting situation where it hasn't already been hammered by light and is potentially sort of primed to be particularly valuable to the organisms, the microbial communities in those lagoons. One of the things that our early sort of calculations have shown us is that although that groundwater flux can only count for a small percentage, less than 5% of the water flux during the summer, it may account for up to 70% of the DOM flux into these lagoon systems. That we're focusing on summer here when river flow is relatively low and the other inputs become relatively more important, but there's an interesting seasonal dynamic there to think about. We also have some evidence that below the sort of, in addition to having superpermafrost groundwater, there are thawed areas deeper down that connect the lagoons with areas of thawed areas under the land that may be interesting areas for exchange we hadn't thought about. Time here. So in terms of coastal erosion, this is work that Craig Tweedy is leading the charge on. He's working on planimetric and volumetric transfer of material from land into the lagoons, thinking about mechanisms and drivers, and of course leveraging previous data sets and efforts and building on those. And he's working on both lagoon specific flux estimates or erosion estimates, and also on sort of domain wide or coast wide estimates along the BLE research domains. We're also sort of along with those just bulk erosion efforts, we're thinking a lot about what's in there. And both in terms of quantities of carbon and nitrogen, for instance, and how those bulk erosional fluxes translate into biogeochemical fluxes, and we're thinking about the liability of that material. I'll move on from that. And the last thing I wanted to just spend a little bit of time talking about, so that was on the input side. Now I'm sort of from the lagoon side. This is just sort of a first look at seasonal dynamics of dissolved organic matter concentrations within the lagoons. This is data from the eastern side that started pre-LTER and bled into the beginning of the LTER period. But the main thing here, without getting into details, is that you can see, in blue is the breakup period. So it's right after the big freshet has happened, big runoff pulse has happened from land. The gray is the summer and the yellow is ice covered. But so those inputs translate into big differences of what we see in the lagoons. And then on top of that, by doing, looking at compositional indicators like SUVA and CDAN ratios, spectral slopes, we get an idea of the sort of processing and transformations that are occurring as well. And there's also an interesting seasonal dynamic to that. From that, we developed a sort of a conceptual model that involves, this large pulse of terrestrial input that dominates during the spring, not just, sort of inundating the lagoons, but in fact, filling flooding out into the coastal ocean more generally. And really, at that point, physics dominates and the terrestrial signal dominates. As we move into summer, one of the interesting things that has emerged from our sort of looking at salinity concentration relationships is that there appears to be net production or net generation of DOM within the lagoons during the summertime. I've had probably associated, certainly that we can see that it's associated with fluxes from the sediments. We think the ultimate source of that organic matter is probably from the coastal erosion and particulate inputs that come in that system that really drive that. And then under the ice, when after the long winter, you still see a considerable sort of buildup of DOM under the ice as things continue as DOM continues to get generated over the winter time. All right, I got 2 minutes. So so that conceptual model was developed based on the eastern sort of view of the world from the eastern node. But as we move over and start working in other places, it's interesting to see that during the spring, concentrations across this west to east area are generally higher in the west than the east during the spring. But by the time you get into summertime, that flips. And so, and what we think is going on there is that this interplay between the inputs and the ocean exchange characteristics and how much and how thoroughly the lagoons can get flushed out from the ocean side determines that concentration point, if you will, where that we encounter during the summertime. Lots still to learn there, but I think it's an interesting sort of interplay between the input side of things and the ocean exchange and indeed the in situ processing side of things as well. And last but not least, then of course, these changes in the physical environment to the runoff, the ice cover, et cetera, and the changes in the chemistry lead to changes in biology. This is just an example on the left of the of bacterial communities, which are distinctly different between the different times of year within these lagoons. The green being June, the light blue being April, and the dark blue being August. And we see dramatically different sort of levels of processing and shifts from net respiration to production as we move through these different seasonal periods as well. We also see shifts not only in who's there and what they're doing, but just the food sources that organisms that live year-round there are using. And you see here a shift from more sort of marine-derived organic matter sources being used in the winter and breakup periods, and greater contributions from the terrestrial side making into organism tissues as you move into the summertime period. And with that, I guess I've burned up my time, but in general, I would say with the LTER, there's certainly lots of data sets that could contribute to a synthesis effort. Some of these are legacy data sets that are pretty beefy already. Others are just beginning to emerge, but I think where there's no data, even a few points make a big difference. And so I'll leave it there.
That's a very good point. Thank you so much, Jim. That was great.
Mm-hmm.
And next is Atsushi Matsuoka from the University of New Hampshire, who will talk about the MALINA and Nunataryuk projects.
Hello, everybody. Can you see my screen?
Yes.
Okay, so I'm gonna talk briefly about the overview of the previous or ongoing Arctic project, particularly MALINA project, which was a France, Canada, USA joint international project, and also ongoing European Horizon 2020 Nunataryuk project. So both projects focus on the southern Beaufort Sea, as shown in the red circle here. So here is Alaska, Russia, Greenland, for those who are not familiar with this location. So the reason why we focus on this area are listed here. So climate change is already affecting this region in terms of sea ice decline and permafrost thaw. And Mackenzie River, which is the largest North American site, carries a lot of the organic matters, partly originating from the permafrost thaw. And this area also covers the large dynamics of the trophic condition, from eutrophic condition in the river mouth to the very oligotrophic offshore near the sea ice. So this area is one of the model study area to examine the impact of climate change on the biogeochemical processes. So the MALINA project, the main objective of this project was to understand how biodiversity and biogeochemical fluxes are controlled by light penetration of the ocean and how they are affected by ongoing climate change. So this project produced in total 64 peer-reviewed publications. So it is impossible to show all of these within the only 15 minutes. Rather, I'd like to highlight three important key processes, which are primary production, bacterial activity, and organic matter photooxidation, which are light-driven processes, so has an important implication for ocean color remote sensing. So first, I'd like to talk about autotroph versus heterotroph, because this is important, has implication for the CO2 fluxes between the ocean and the atmosphere. So this figures was presented by Ortega Lutete et al. 2012. The color represents the ratio of bacterial carbon demand to the primary production. So if the value is higher than 1, it means this area is a heterotroph and vice versa. Of course, this ratio is influenced by the primary production and the heterotrophic bacterial activity, which are also influenced by quality and quantity of organic matters, so bioavailable versus refractory organic matter. So the author also examines the relationship between the bacterial production and different sources of organic matter. In this case, chlorophyll and upper panel, and the lower panel represent the relationship between BP and larval organic carbon. In this case, total dissolved amino acid. So relationship vary depending on the quality of organic matter. So this is important to determine the autotroph versus heterotroph conditions. So I talked about the autotroph versus heterotroph condition for the coastal waters. Now I'm talking about this condition for the offshore area, specifically talking about nitrogen source in the oligotrophic waters. So in the next slide, I will show the result along this transect in the western transect, which is a major discharge area of the Mackenzie River. So this panel represents from the top to the bottom, nitrate concentration, pico-eukaryote or pico-phytoplankton, and nano-phytoplankton at the bottom. What we see here is that although nitrate concentration are very low at the surface, we observe abundance of the pico-phytoplankton almost at all stations, meaning that nitrate was never depleted completely or never reached below the detection limit. This provided important hypothesis that bioavailable nitrogen, but particularly ammonia, can be photochemically produced to the surface waters. It's called photoammonification. This hypothesis has already suggested by previous papers, but our result also suggested this hypothesis. So the Wishenshi et al. 2012 examined this topic by bringing the samples during MALINA cruise back to the laboratory, and they used the solar simulator to incubate the water sample to see if the photoammonification occur or not. This is one of the results. So clearly we observed the photo production of ammonia over the course of the incubation. So the question is how this photoammonification influences the biogeochemical processes. So using the numerical model, Fue et al. 2013 examined this topic using this biogeochemical coupled with physical model, as shown in this schematic diagram. On the left-hand side, I'm showing you the modeling result in the black line and the observation in the red. For the primary production, bacterial reproduction, nitrate plus ammonia uptake, and ammonia regeneration. So without photoammonification, we clearly see the difference between the model and observation. But if we took into account this photoammonification, the model reproduce much better relative to the observation, although we still see the significant difference for the bacterial production. But main point is that the photoammonification is very important to better understand biogeochemical processes, especially in the oligotrophic conditions. So this study also suggested that this kind of diagnostic approach is very important for a better understanding of the processes. So now I switch to the another large project, ongoing project, Nunataryuk. I also point out that this project not only focus on the physical science, but also social science. For example, how this permafrost thaw impact on economy, infrastructures, and the human life and so on. So we are involved in the one work package 4, which is coastal waters. So one of the main objectives of this work package is to better understand seasonality and the fate of organic matters. So to achieve this objective, we use different platforms, including the helicopter landed on the ice, or even hovering helicopter, from which we deploy the Niskin bottle to take water sample from the surface, as well as optical instrument deployment from the hovering helicopters. And later in the season, we use a small boat as well. So again, we pretty much focus on this coastal or even delta. So most of the station, over 90% of the station were located less than 15 meters. So we focused on the delta to see the seasonality of the organic matters in this area in the McKinsey Delta. Although many results are coming up, I'd like to highlight one of the published results in terms of the impact of different organic matter on the microbial community composition. So what we did during this study was that we collected the soil samples from very different locations. For example, fluvial condition, lacustrine condition, and moraine condition. So we took the soil samples from both active layers as well as permafrost. We mixed soil sample together, so which are used as organic matter sources. We also took the water samples from the ocean, close enough to the Mackenzie Delta. So this water was used as a bacterial inoculant. So basically what we did is that we fed these different sources of organic matter to this bacterial inoculant to see how the microbial community respond to individual organic matters using chemostat experiment. So this is one of the highlights from this study. So left-hand side, we showed different communities of the bacteria prior to the experiment. And right-hand side, I'm showing you the relative abundance of the communities. So one of the highlights of this study was that especially for the moraine organic matters, including the permafrost, we clearly see the significant increase in the gamma proteobacteria, which is often used for the biodegradation experiment. And this has ability to quickly use larval source of organic matters. So this is what was observed during this experiment as well. So clearly, the organic matter from different sources influence microbial community composition, which has important implications for the coastal erosions and permafrost soil on the coastal processes. So to summarize, the Southern Beaufort Sea was mostly heterotrophic condition in the August 2009 in particularly, but this relationship vary, of course, depending on the season. In terms of remote sensing, this can be influenced by the, can be seen by the prime production. So this is a common challenge at the lower latitude, but still it's very important to accurately estimate primary production and even chlorophyll concentration in the river influence coastal waters. So this is a still challenge at the Arctic region as well. The content of dissolved organic matters influences bacterial production and the community composition. So this has several important implications. As I mentioned earlier, the different sources of organic matters might significantly influence for the microbial communities and coastal water processes. So this can be influenced by the, for example, the coastal erosions or permafrost inputs. I'd like to also point out that the better characterization of organic matter is very important, particularly sources, composition, and bioavailability. And also important to note that some of the organic matter has no color. So obviously we cannot detect this kind of organic matter from space. So this is something that we have to keep in mind. Seasonality, as Bob mentioned earlier, that there is a significant difference of the between the Yukon and Mackenzie River in terms of the distribution of permafrost as well as river discharge, and how this seasonality of organic matters impact on the coastal water system, we don't know very well. So definitely seasonality is one of the important topic. And priming effect. So this can be very important particularly when we think about the photooxidation of organic matters, which might stimulate the microbial community for further decomposing the refractory DOM. And we don't know very well about priming effect for the Arctic region. So this is another topic to be considered for the Arctic color project. So finally, diagnostic approaches that I showed as a numerical model or even a chemostat experiment is very useful for better understanding the processes, including nutrient dynamics, organic matter processes, and even useful for future prediction of phytoplankton phenology and many others. And finally, Antonio asked me to show the availability of the dataset. So MALINA dataset freely available, published by Philippe Mascot Edward 2020. So this is the website where the data are available. So please check this website if you're interested in this dataset. Regarding Nunataryuk project, this is still ongoing and partly influenced by COVID pandemic. Many of the analysis have delayed. So unfortunately, at this point, these data are not available yet, but I will keep you informed. And it would be great to integrate with pre-Arctic cruises data set to better document the organic matter processes and beyond. That's all. Thank you.
Thank you so much, Atsushi. That was really great. So it's amazing. We are on time. Now we have 15 minutes for questions to all our speakers. Again, Anne, Rob, Jim, and Atsushi, thank you so much for your presentations. These were great summaries. Any questions for our speakers? And feel free to unmute yourself and ask the question directly to the group. Jim, I have a question for you. That was a great description of the NSF LTER program. And it's great that as part of this program, you're trying to integrate observations with new biogeochemical models that really link the terrestrial environment to the aquatic environment. I was wondering, were there any changes that you felt you needed to make to your measurement approach guided by the model simulations, the biogeochemical model simulations, or any specific parameters that turned out to be particularly important for the model parameterizations across this very complex environment, the lagoons and the aquatic sites across the Alaskan North Shore? You're muted, Jim.
Sorry about that. Yeah, I guess two quick answers to that. One is we're starting to get some sense of that, but we're so early on in terms of the core LTER data set development that it's hard to say. If you asked Mike, he would say, sample the rivers more in more places, right? One could spend, I did have a whole project once where all I did is sampled rivers over and over and over again on the North Slope, but that was a big job in and of itself. So, but obviously more in terms of capturing dynamics, having little snapshots is challenging, and you have to wait an awful long time before you build up enough data when you're taking little snapshots to start to be able to put the picture together. So, and Yvette Spitz, who does the biogeochemistry modeling on the ocean side, and I didn't get into, but she's making great progress on that. would say the same thing. She just needs more. It's not, you're not collecting something I need. It's that I only have 3 points and I need, 300. And so it's a question of data density, I guess, and sample frequency, and more is better, I guess, from the modeling standpoint for really understanding how well the model's performing, right? If you only have to make it match at 1 point in the summer, that's different than making it match, a summer's worth of points.
Yeah, that's a very good point. So maybe something that breakout groups could address also during their discussion. How can we do that? How can we improve in terms of the frequency and the temporal coverage of our measurements, capturing different seasons as well, right? Yeah. And having more frequent measurements.
Yeah, and I guess I would just add to that, related to that, for Arctic GROW and for LTER, when you're working on sort of longer term projects and you have the luxury to go back again and again and again, you are building data sets up over time, but it takes a lot of patience, right? And so you're right, some targeted efforts at higher frequency ultimately are hugely helpful. I think Mike would certainly say, yeah, for seeing some of the, because if he's modeling things on a daily time step, how unless you have some data sets on a daily time step, it's hard to know how you're doing. So there's value in those more focused, even if they're short term, those more focused efforts.
Right. Okay, so, great.
This is Patty. I have a question or comment for Rob. He made what I think it's a fabulous reminder, and I wonder whether you can expand on that, especially for the oceanographers. You said, don't forget that Flex 2 estuary is different than Flex 2 ocean. And could you expand on that?
Yeah, absolutely. And again, apologies for me. I think I'm slowly losing my voice today, so I might not be with you tomorrow at this rate. But we have a good amount of data, I think, from a whole host of projects, some of which was just presented at that, I don't want to say that it's always at the head of estuaries, but it's pretty close. It's to the last downstream gauging station in a lot of rivers across the Arctic. There's some data that exists. There's good data that exists in coastal ocean, often in late summer, sort of July, August, but not very much, but there is some. But there's a real dearth of data, going from fresh waters out across estuarine environments into, well, certainly anything like full salinity. And then there's the secondary problem that Jim's last question was just touching on in terms of, well, what time step of resolution do you want that in? And in these highly dynamic Arctic systems where that's changing hugely in late May, early June, there's really no data that I'm aware of, or very, very little. And then there's a little bit later into the summer where it's easier to work there. I'm not going to say it's easy, but it's easier to work there. And now the question becomes, of all those times, which is the most important, right, to address that question? And unfortunately for us, I think one of those most important times is across that spring freshet period, where we really have very little data, and it's changing very quickly. If you go from last week of May through to the last week of June, early July. And so targeted campaigns across that kind of time scale, I can see getting at what Jim was just talking about would be very helpful, probably very helpful for the modelers. And the environment that you're examining is changing greatly. I mean, I tell people the stories where we'd sit out on the Yukon and do daily sampling, and I'm sure everyone's got similar stories, but the surface temperature of the river's getting warmer by 1 degree Celsius a day for 14 days in a row. And so when you think about the impacts there on things like heterotrophy and the rest of it, photochemistry as well as a big player starting to come in as we lose the ice, it changes very quickly in a very short time period. So I think To answer Paddy, your question, we need to do a lot of work, which is what Arctic Colors is trying to do across the land-ocean interface. There's just a dearth of data there generally. And it'll be very interesting to see in the next couple of days about other data sets that exist and how many there really are. But then it's also the timescale of this, where there just really isn't much apart from in late July or July and August because of the logistical side of operating up there, probably at the period where there's the most happening. Concurrent with spring freshet. And so does that get at answering two questions in one there?
I would like to chime in on that one, Patty, a little bit too here and just point out that on the Yukon River, Arctic Grow measures at Pilot Station, which is 120 river miles from the mouth. So, 120 miles for going through the Delta for who knows what to happen. And so I think that's also kind of fits into that idea there that, you've got a 120 mile bioreactor that we don't really have accounted for very well at this point.
Yeah, it's an unfortunate disconnect, I'd say, between where funding's sort of available and this land-ocean interface has maybe suffered as a result that there's not been as many studies as I think as a community we'd like across it, because it's not fallen under traditional funding boundaries, if that makes sense.
Yeah, but at the same time, it is an absolute pain in the to sample. Okay, there's there are logistics, the safety issues. I mean, Jim knows the access issues. It's just not that you're going to drive. I remember saying this, that you're just simply going to drive to the dock and then get there. And I said, where is your dock? And I'm going to just take the pickup. And I go, and where are you going to get the gas for the pickup? And so that's a reality that we discussed tremendously over the issue of the science plan, the wishes versus the realistic component. And that's one that especially those of us here today who are not familiar with that, please ask and don't take anything for granted, which is why I thought this flex to estuary being different from flex to ocean is just such a beautiful way of putting such a difficult issue. Thanks to both of you.
There is one question in the chat box. The photochemical by Paula Wamsley. The photochemical production of ammonia is very interesting. Do sky conditions, specifically cloud conditions, have much impact on the production rate?
Yeah, that's a good question. So I said yes, because our result was obtained using the incubation using solar simulators. So one of the issues, as you pointed out, is how this impact really on the natural waters. This is challenging, but yeah, you're right. We have to think about this natural condition, blue sky versus cloud cover in terms of the diffuse and direct component of irradiance.
I have a question, Maria.
Yeah, Joachim, go ahead, please.
Yeah, I was wondering whether trace metals are actually an issue in the Pan-Arctic region, because I didn't see it in the list of measurements, core measurements that were made. And was it deliberate because they're not important?
Joachim, this is Patty. No, it's not an issue of not important. It's an issue of that we had to do a prioritization of the variables and parameters to be measured. This was a very strong request from the review panel. We needed to focus. And so it's a very different environment given the enormous terrestrial input. There are folks in in the audience right now who know more about this than I do. Very different from the open ocean, where no geotraces Arctic is probably the most recent snapshot of what's out there. And that's a very different system from this shallow, seasonally very active and very terrestrially influenced system.
Thanks, Patty. I have one last question, and this is concerning HABs, since we are talking about change. And one of the issues that often comes up is whether there'll be a migration of toxin-producing HABs into that region, which is, of course, of relevance to the local communities. And I wonder whether any of our previous speakers have come across this in the Beaufort Sea, for instance, along the coast.
I'm sorry, Joaquin, could you repeat? It is a change in, what did you say? I couldn't understand.
I was trying to understand whether we have this problem of HABs.
HABs.
Yeah,
oh,
toxin-producing harmful.
Oh, harmful algal blooms. Okay, yeah. Thanks.
Don Anderson, I know, is dabbling at least in that a little bit around the Barrow area, and identifying. I wouldn't say he's seen blooms, but they're the more they look, the more they find in terms of HAB organisms. The question is, what? Yeah, do they have the potential bloom? Are they, what are they doing? What are they? Are they actually toxic? Right? They're not actually toxic all the time. But it's an area that at least some people are starting to think about.
Yeah, I see Jackie's raising her hand. Go ahead, Jackie.
Yeah, I would just say there's a new PNAS paper out that I'm on with Don Anderson, and we are having blooms of Alexandrium, and we have above toxic of EPA levels in the clams that we've been collecting. And so these are off of Ledyard Bay into the Chukchi, but off to the east of Barrow Canyon too. So they are a big concern by local communities, and we are seeing them in the food webs of parts of the Chukchi. So, and they're cysting. So the blooms are actually going down, they cyst over and they're sitting in the sediments, and the water's warming up and they're now blooming. So it's Don gave a great talk at the Bering Strait just this last week, and it's a recording you can look at out of the Nome area.
And Joaquin, the Canadians, federal scientists started to sample HABs along their coastline long before anyone was funded in the US to do that in a consistent manner. But I do not believe that their data are out. They may be available. I don't know. I haven't checked, but they started a few years back already.
Thanks a lot, Jackie and Patty.
Okay, great. So it's already 2:35, and thank you so much for the questions. But it's time to move on to the breakout group discussion. Let me describe a little bit for a minute how the breakouts will work. You will be assigned randomly to a breakout group, and all breakout groups will be addressing the same questions. Some of the meeting organizers will join breakout groups to brainstorm with you. The link to the Google document with all the questions is provided in the chat box, but I will also be sending the questions to the breakout groups in the chat box after everyone joins their group. For each group, I would like to just ask, please designate a person who would take notes and report back and communicate your answers to our whole group during the whole group discussion. We will be spending something like 3 to 5 minutes reporting back. Some groups are going to be answering some questions. I think there is a way also to add your comments, notes directly to the Google document with the questions. Heather, if you can let us know if this still is an option on the Google document. But if you want to take notes outside the Google document, please feel free to do so. As you will see, yeah.
Everyone has editing privileges on the Google Doc.
Perfect. Thank you, Heather. As you will see, the Google document has 3 main questions. The 4th question is mostly relevant to the modeling efforts and the satellite data sets, and we will be discussing this tomorrow and on Wednesday. Please try to spend, do not spend more than 20 minutes on the first question so that there is time to address the other questions as well. So with that, let's move to the breakout groups, unless if someone has a specific question to ask. And please, if we don't have time to discuss all the notes and capture the discussion you will have within the smaller breakout groups, please send us your notes to this email address, and I will put it in the chat box as well. Thank you.
So I don't think it will move the organizers, who are my co-hosts. You will be able to choose your own rooms. And if you look in the Google Doc, I have put your names next to each of you has a room so that you can divide yourselves up.
Great, thank you, Heather.